none better than Sarah. She's just been amazing and a very good colleague and friend as well as advisee. And I've never done this before, but I realized that I've got to write everything down because usually I just kind of wing it because I think I can remember everything and I inevitably forget something. So I just want to give you kind of a brief on her background. So she got her bachelor's degree from Oregon State in environmental science and also worked in Astoria for a watershed council there and implemented a trail, I believe, in Astoria back around 2007 or 8.
Back then, yeah.
And so she already came to Portland State with experience as well as education. Started her master's degree and did a master's project out in Coos Bay, where she looked at the effect of grazing on reed canary grass and other invasive plants in the riverine, riparian communities there, plant communities, and found that intermediate levels of grazing were helpful in suppressing non-native plants. And so grazing actually benefited native plants and published that in a peer-reviewed journal. And then in 2011, she was accepted in our first IGRIT cohort class, and that was a really hard, there was a large pool of applicants for this, and so Sarah was one of the few who were selected into that. And she immediately was also part of the Radboud Honor Society, or Honors Academy, I mean, and went to Holland, worked with Dutch and Indonesian scientists to look at sustainability issues in riverine delta areas. She further became a GK-12 fellow here in ESM and has had numerous other grants in addition to those 2 fellowships, very prestigious, both of them, IGRIT and GK-12. She benefited greatly from National Park Service collegiality and interaction and mentorship in Astoria, and also received a nice grant from them, as well as a lot of in-kind support. She worked with the Cowlitz Tribe and with many other entities in the Astoria, Lower Columbia area. So she, and LCRAP, BPA, the more I stand here, the more I remember. There's many players that she's worked with to be able to do this work. She's also collaborated with social scientists as well as natural scientists to look at some of the issues in what has begun to be called the restoration economy of Astoria. And Sarah's played a role with several other IGRIT and other students and faculty to try to tell that story. So it's just really been a wonderful time to work with her. And the bittersweet part of a dissertation and a really good graduate student is, that time's coming to an end. So I won't cry or get too, sappy here. But at the same time, it's also a great thing, right? When a doctoral student matures into a PhD and a practicing scientist under her own power, with her own independent ability to create research and to go forward into the future. So it's just a great honor to present her. And I also want to recognize, before I do that, her committee. And so they're all sitting up in here. So Drs. Martin LaFrenz, Andon Pan, Sarah Epley, and Jennifer Morse are all here and have given her good support along the way as well. So with that, Sarah.
Well, Alan, thank you so much for all of those kind words. That's kind of hard to follow that without being emotional. It's also really hard not to be emotional when I see a room of all these people that I love and who helped me, kind of get to where I am today. So I'm going to try to wrap this emotional part up so I can my research. But it's 9 a.m. It's early. I'm very appreciative of my committee for being able to make the time to help me today, and for all of you and all of your support through the years. Family, friends, so many interns. So thank you so much. And so without further ado, I will present my research on ecosystem recovery in Oligohaline tidal wetlands of the Columbia River Estuary. So first, I'm just going to give you a little background information about my study area, and then I'm going to go in to some of the dissertation objectives, talk about the 2 studies that I did in detail, and then some just general conclusions. So for those of you who are not familiar with the Columbia River Estuary, it runs from the Bonneville Dam all the way to the Pacific Ocean, and this whole reach of the Columbia River Estuary is tidal, and it's a fairly fresh water estuary. And so here at the mouth of the estuary, this tidal wetland complex is considered Oligohaline. And that just means that it's a little bit salty. So it's not a freshwater system, but it's also not a brackish or saltwater system. And so this is really interesting because this is a very critical area of the estuary because it provides habitat for salmon as they are transitioning from ocean into fresh and vice versa. And so this is where my research takes place. And so Young's Bay has a really interesting story of land conversion. So if you see this map here, you can see all of the extent of green. That is the historic extent of tidal wetlands and tidal swamps in Young's Bay. However, if you look here, this is the current extent of tidal wetlands and swamps. You see that there's really very little green. And this is because there's been a 97% loss in tidal wetland habitats in Young's Bay since the 1880s. And this land conversion has taken place mainly in the form of dikes. So they put large mounds of earth, they separate these areas from tidal flooding, they become drained, and then they use them for agriculture. And in Young's Bay, the primary form of agriculture has been grazing and dairy farms. By 1914, there were already 50 dairy farms in Young's Bay. So, a very, very quick land conversion occurring here. And so this land conversion in Young's Bay and this loss of tidal wetland habitats is actually not a unique story. This is happening throughout the Columbia River Estuary and really throughout the estuaries of the world. And if we think about the total loss of wetlands, not just estuaries, we've lost 87% of our wetlands since the 1700s globally. So this is an important thing to think about. And so what does this look like on the ground? Well, if you are in Astoria and you're up in the column, which is right here, and you're looking down on the entire watershed, what you see are all these beautiful low-lying green areas that are used as grazing pastures. And these all used to be historically those tidal wetland habitats. So just a kind of a visual for what that looks like. And so why do we care about these tidal wetland habitats? So what we've realized is that through their loss, we've also lost many critical ecosystem services. And here in the Pacific Northwest specifically, the loss of these habitats have been directly tied to loss in salmon fisheries, in addition to having all of those dams in the Columbia River. So loss of tidal wetland habitat and then blockage of habitat from the dams have been the 2 leading causes of a loss of these endangered salmon species. And so in an attempt to recover these lost habitats and recover these this salmonid critical habitat, we've done a lot of restoration efforts. And so here's a zoomed in photo of Young's Bay. I'm going to show this again later, but I just want you to see all those dots. That's pretty much all restoration projects. So there's been an extensive amount of restoration in the lower Columbia River, particularly in Young's Bay. And really, this restoration approach is happening all over the world, and it's called tidal reconnection. And it's where they go in and they remove a portion of that dike or that levee, and they allow that tidal flooding to come back into the site. And the idea is, by reintroducing that tidal flooding, that the site will, over time, regain all of that habitat structure and become sort of a natural reference wetland again, providing all those ecosystem services. And so, underlying this idea of restoration is this assumption that over time, after removing a disturbance, that the site will regain those historic habitat conditions. And this is what I call the theory of ecological restoration recovery. And it's embedded not just in tidal reconnection efforts, but in all restoration. This idea that if you remove a disturbance, that the site will then take a journey back to its historic condition. The issue with this assumption is that because restoration projects are seldomly included in research and monitoring, we really don't have a lot of data to support that this is always occurring. And so I found this as an opportunity for myself to dig a little deeper to see what's really going on in these wetland habitats, specifically in these Oligohaline habitats in Young's Bay. And so thinking about all of the known drivers of wetland plant community and ecosystem development within these sites, I was able to sort of make some targeted investigations. And so I'm going to show you this. This is a lot of information, but what I want to highlight is the major things that change in a site at the time of restoration. And so you have, all of a sudden, you have tidal flooding. So, a site that was dry because of these dikes or levees is now being tidally flooded. So you have a change in the frequency and the duration of flooding, and a change in the salinity conditions of that site. This is going to lead to a change in the soil conditions. Lower soil oxygen levels because of the flooding, also increase in salinity, and then these are going to result in some differences in the nutrient dynamics on the site. Now, this shift in both the tidal flooding and the soil conditions really creates a template for wetland plant community development. And whether there's invasive species or a lot of native species available on the site, this is going to then provide the outcomes of that restoration. So all of these drivers are going to be working together to get our restoration outcomes. So using this understanding of wetland habitat development in these restoration sites, I developed some questions that I wanted to ask in my research. And so for my first study, I wanted to understand if reference or natural tidal wetland conditions could be detected recovering over time in these sites. So specifically looking at soil conditions and plant community characteristics. And then I wanted to see if there were any observable patterns in plant community development within these sites based on this restoration gradient that we created by changing the tidal flooding and salinity conditions. And then based on the observations that I had from this study that I did looking at these patterns, I decided to take a deeper look into the seed bank composition of the native and non-native plant communities. And so this is really important because when we are doing this passive restoration approach, we're literally breaking down the barrier to tidal flooding and then just allowing the site to passively restore. And so you're really depending on seed dispersal from nearby wetlands to create that template for wetland plant community development. So I wanted to know what was going on in the seed bank. And then I wanted to understand how the seeds in the seed bank responded to changes in tidal flooding and salinity conditions within these sites. And this is critical because if we think about how we are going to adapt our restoration approach, we may be changing those conditions on the site. Additionally, thinking about climate change and sea level rise, how are these conditions going to change the plant community dynamics on these sites? And so focusing right away, we're just going to jump into study one, looking at, do we see changes in plant community and soil characteristics over time, and then are there patterns within the site looking at plant community characteristics? So here's that map again. So we have the Columbia River up here, and then Youngs Bay, and then we have three major tributaries, the Lewis and Clark, the Youngs River, and then the Luce River. And you can see that the sites are scattered throughout those. So I had 11 restoration sites, and these ranged in age from 1 to 54 years. And those numbers here, 25 to, that is the age of the site at the time I surveyed it. So how many years since that tidal reconnection took place. And so I had 11 of those sites, and then I had 4 reference wetlands. And you can see that these are sort of along a gradient on the Youngs River. Now, reference wetlands are sites that have never been diked and drained and really represent sort of the natural tidal wetland condition within Youngs Bay, so the ideal or goal condition. And then I had 2 pasture sites. And one of these pasture sites, the Cowlick site, was actually very continuously grazed up to the point that I was surveying it. So it's an active pasture site, and now it's slated for restoration at any time. So it was, pre-restoration conditions. And then this other pasture site had not been grazed for 8 years, but it had not been tidally reconnected. So it was interesting to get a look at these pre-restoration conditions and how they may vary. So using all 4 of these sites, I was able to develop a space for time approach to look at recovery over time, using these sites as sort of that space for time. So this is called a chronosequence. And what I want to point out right here is that along this axis, here, this x-axis, you can see a 0 to 54. And then all of these highlighted numbers are sites that I had along that chronosequence. And so this approach, this chronosequence approach, is really common in wetland science and soil science, tracking changes over time. And I developed hypotheses to determine at what point along this chronosequence, which sites would I expect to see recovery to reference conditions. And so this literature that I pulled from, I focused on tidal reconnection projects as many studies as I could find that looked at recovery after tidal reconnection. And there really weren't that many, and there were no Oligohaline studies. And so, this gave me sort of a loose idea on, when I might expect to see recovery based on these other studies. And so I'm going to jump right into those. But first I want to point out that here on my chronosequence, I have a lot of sites that are between 0 and 9 years, but I do not have a lot of sites between 9 and 54 years. So I just have a few scattered. And this is because restoration did not really become popular and was not well funded until about 10 years ago. And so you see that it becomes, I have all these sites in here, and this is really when restoration started becoming a thing in Young's Bay. Now this 25-year site was also a restoration site. It was the oldest tidal reconnection site in Young's Bay. However, the 34-year and the 54-year, these are accidental restoration sites. However, the approach to restoration was very similar, because what happened is that during a storm, the tide gate and the dike blew out, and it was never replaced. So this tidal reconnection happened, it just happened naturally. And then the landowner just never fixed it. And so that's something to understand about that chronosequence. Now jumping into the hypotheses. Now when I'm thinking about plant community composition, I'm going to be focusing on native and non-native cover of these species, and I'm going to be thinking about species richness and overall plant community similarity to these reference sites. And so what I expected, based on the literature, is that I would see recovery to reference conditions within 5 to 10 years. And so I should point out here, this is relative equivalency to the reference sites. So whatever parameter this is how it compares to those reference sites, and here you can see the reference range. And so here we see within 5 to 10 years expecting that plant community shift. Now for plant productivity, which I'm focusing on native and non-native plant height and then overall biomass, looking at between 4 and 14 years to recover. And then moving on to soil conditions, soil development, focusing on soil compaction and soil organic matter content. These in the literature were looking like they were going to take a really long time to recover, greater than 20 years. And so you can see here that I'm anticipating those older sites to be recovered, but those younger sites not. And then for soil nutrients, focusing on nitrogen and phosphorus, I expected it to take anywhere from 3 to 5 years to show a decline in these nutrients, them being elevated towards reference levels. And I anticipated that they were going to be elevated compared to reference conditions because of this active grazing and the manure spreading that happened for 100+ years on these sites. And so my fifth hypothesis is really focusing on plant community development within the sites. And based on my knowledge of the ubiquitous abundance of reed canary grass throughout the lower Columbia River, and I anticipated that there was going to be a gradient from sort of the low marsh, so that's going to be more frequently flooded than the high marsh, which is going to be much less frequently flooded, of increasing non-native abundance, primarily reed canary grass. And this is because we know that reed canary grass likes wetland environments, but it doesn't like it really wet. And so sort of based on that information. And so to summarize this, I hypothesized that the low marsh areas were going to be much more similar to reference conditions over time than the high marsh areas. And I anticipated the high marsh areas may sort of become invaded by these non-native species and be very resilient in that situation. Okay, so jumping into the methods. I have these 11 restoration sites, 4 reference sites, and 2 pasture sites. I surveyed half of them in 2013 and half in 2014. Some of you know exactly what that looked like because you're out in the field with me. This was a lot of effort. We collected over 254 soil samples and biomass samples and over 1,000 vegetation quadrats across all of these sites. I also collected water level or water elevation data and salinity data across a subset of sites. And this was so we could understand if there was any kind of tidal flooding gradient that I needed to be concerned about. And what I did not find that there was a significant difference in the amount of tidal flooding happening here on the 9-year site compared to any of these in Young's Bay. So that was really good because that means these were all comparable in terms of the amount of tidal flooding they were experiencing. And so this took over 900 hours personally of field time. That's not including my intern's time, that's not including lab time or data analysis or writing. So that was a lot of effort in the field. And so this, here, let me go back here. I want to show these 3 sites here. That's the Lewis and Clark National Park. I'm going to show you a map of those sites so you can understand what this looks like on the ground, these transects and vegetation sampling. And so here, I've turned it on its side. We have the Lewis and Clark River here. This is the 54-year site. the 1-year site and the 6-year site. And what you'll notice is they're all very close to each other, but they were tidally reconnected at different times. And I distributed my transects randomly across the elevation gradient of these sites. I collected vegetation quadrants every 5 meters, and then every 20 to 30 meters I was collecting plant community and soil data. And so jumping right into the data. So this is a real, Alan wanted me to make sure that everyone understood that this was not a photoshopped photo of me in the field collecting data. This is an actively grazed pasture in Young's Bay, and I am collecting RTK data, which is elevation data. And so these are agents of soil compaction. So soil bulk density is a measure of soil compaction. These sites have all been grazed before restoration, about 100 years of this. So we're expecting the soil to be very compact. And so let me orient you to what this graph looks like, because all the graphs I'm going to show you following this is going to look very similar. So here we have soil bulk density. On the bottom we have 0 through 54. That is the age of the sites. The sites that I have data for are highlighted. You can see the 6-year site. I have 2 data points there. 0 is the pasture sites, and then here on the end I have the reference sites. And so there are 4 reference sites. Here 2 of them are overlapping, so it's harder to see. And then I'm going to have the mean range of the reference sites highlighted so you can kind of see when these each site falls within that mean range. And so what you'll notice right away is that we did see a decline in soil bulk density over the chronosequence, with recovery observed within 3 to 9 years. And so this was really interesting because it was a little bit faster than other studies had noticed. And so now moving on to soil organic matter, and I do want to point out that Drew is not actually sampling soil organic matter here, but he is very stuck in the mud. We were collecting some equipment. So again, soil organic matter expecting to take greater than 20 years to recover. And so again, we have organic matter here, we have the sites on the bottom, and then the mean range. So we did see some variability in organic matter among the sites, but overall there was a trend of recovery, with recovery occurring between 3 to 9 years, so very similar to the bulk density. Now looking at native species richness, again predicted within 5 to 10 years. And what we found here was, first let me point out that the yellow here is the non-native species richness, and then the green is the native species richness, and you can see the range of both the reference native and non-native. And so we found that there was a really clear trend of increasing species richness across the chronosequence, and a clear trend of declining species richness, with native species richness observed within 6 years, and non-native species richness, a decline observed within 3 to 6 years. So this fast transition with the non-native species richness is really expected, because these are upland pastures that are not necessarily wet all the time. They have a lot of upland grass species, and so if you flood that, then all of those species are going to, you would assume, die out. So you see that really steep decline right away. And now looking at native and non-native cover, predicted within 5 to 10 years. And very similar to the other graph, we have the native here in the green, the non-native in the yellow. And what we found was that it was actually a fairly clear trajectory. Both the native and the non-native were observed within the reference range within 3 years. However, there was a variability observed in native and non-native plant community development within the sites, and you can kind of see that here. And this really leads to the last hypothesis and the data for that. Thinking about when I broke the data into low, mid, and high marsh, and I did that based on the frequency and duration of tidal flooding you observed sort of at each plant community, what I found was that the mid to low marsh actually achieved reference levels of plant community cover and similarity within 3 to 5 years, and that is looking at lean-bee sedge and bulrush, while the high marsh area up here, it did not achieve native cover. It was actually maintaining non-native cover across the entire chronosequence. So even the 54-year site was dominated in the high marsh, and so this was a really interesting finding. Additionally, in those invaded high marsh areas, we found that the soil pH was much lower than the reference high marsh and the mid to low marsh areas. Additionally, the soils were also not as salty, so there were lower soil salinities. And these characteristics were much more similar to those pasture sites than the reference or restored low to mid marsh. So restoration was just not occurring in those high marsh zones. So summarizing the conclusions from study 1, overall we found that trajectories of plant community recovery were observed, particularly in the mid to low marsh, within 3 to 6 years. Plant productivity, so biomass and plant height, were also observed within 3 to 6 years, following very similar trajectories. Soil organic matter and bulk density were observed between 3 to 9 years. And then really there was actually no trend in soil phosphorus and nitrogen. It was really variable among the restoration sites and the reference sites. And I was surprised to find that the pasture sites did not actually have very elevated nitrogen and phosphorus conditions compared to the restored and reference sites. And then inferring from those observations along the elevation gradient, it does appear that restored tidal flooding gradient, that gradient that we restore, is really critical to native plant community development. Okay, so when I finished that study, after spending all that time in the field, I was really interested in understanding the exact mechanisms driving that native and non-native zonation. And this is really important because if you're thinking about, trying to restore these habitat for, salmon and ecosystem services they're providing, if you compare how those invaded high marshes function to those reference high marsh conditions, so native dominant versus non-native dominant, they are actually providing a really different habitat structure for macroinvertebrates and other species. And it's, it's an issue. And they've actually done studies in the Lower Columbia to show that reed canary grass actually do not host the same variety of macroinvertebrates, which the salmon eat, than the lingby sedge or other natives. And so this is something that's critical. We need to think about, what's driving these native and non-native invasions? And if we understand these mechanisms, maybe we can manage them. And so here's a picture of a site before restoration is occurring. They've cut a channel on this site, which is actually not necessarily common among the ones I looked at, but this is a new site that's just being restored. What I really want to point out is that you can see that not a lot of soil has been moved. The surface of the wetland is not being excavated, it's not being changed, there's no planting or seeding going on. And so we're depending on seed dispersal from nearby wetlands to create a native wetland plant community. And so I decided to jump in and take a closer look at the seed banks of these native plant communities. So the dominant native species are lingby sedge and bulrush, while the dominant non-native species are reed canary grass and common rush. And so I wanted to, like I said, take a closer look at the seed bank. And if you're really confused right now, the seed bank is literally the seeds that are the plant seeds in the topsoil. And seed banks actually have been found to be super critical for wetland plant community development sort of across all wetland types. And they're really important for plant community resilience. So if there's been a disturbance or something, that seed bank is there to sort of sprout up and redevelop that plant community. Seeds are also really cool because seed germination is triggered for each species by a different set of environmental characteristics. And so if you're an upland plant and you like your soil a little bit dry, your seed is not going to germinate in really low oxygen, muddy wetland soil conditions. But if you're a wetland plant and you kind of prefer those and you've been adapted to those conditions, your seed will probably germinate in those sort of more harsh soil environments. So it's really interesting in that way. Okay, so jumping right into the hypotheses and some of the questions for this study, I wanted specifically to understand how seed bank compositions of the native and non-native plant communities compared. So were both native and non-native species equally distributed across the wetland seed bank? Were they found everywhere, or are they more dominant, are they more patchy? And then how do the seeds in the seed bank respond to differences in tidal flooding and salinity conditions? And so this is important because maybe there's a preference going on in terms of germination and plant community development. And so using this essentially 2x2 framework I've developed, I can infer from my results what the major drivers of plant community development are. And so I'm going to try to walk you through this. So if we find that the seed banks are very similar, which means the seeds are being dispersed sort of equally across the wetland high to low elevation gradient, and we find that these species are not showing a germination preference for one area versus another, then we can kind of hone in and say plant competition is the main thing driving this zonation pattern that we see in the field. Now, if we see that the seed banks are very similar, again, really well distributed, both native and non-native across all of the wetlands, but we see a difference in how they're responding to the different tidal flooding and salinity conditions. then these tidal flooding and salinity conditions are likely the major driver of the zonation. So maybe the wetland plants, the natives really like that low marsh condition, and the non-natives really like that high marsh condition, and they show a germination preference. That's the zonation is going to be driven by those environmental gradients. And then, what if the seed banks are really different? And we see a lot of, the natives are really self-seeding, and there's more natives in the native low marsh area, while there's a lot of more non-natives in the high marsh area. And so we have some resilience from self-seeding. However, we see that they are really responding similarly across the elevation gradient, across that flooding and salinity gradient. Well, what this means is that they're happy to grow in any of those zones, but where they land, where those seeds are most abundant, they're probably going to get an opportunity to grow first and then shade everyone else out and kind of have a leg up. So that initial establishment, who's there first and how many, and then plant competition is going to be driving that zonation. And so what's really important to understand about seed germination of these species is that a little bit of shade is going to suppress germination completely. So if you just happen to sprout up and shade out your neighbors, they are not going to be able to grow. So you're going to have a leg up in terms of being able to develop and outcompete them. Okay, and then lastly, if you have a really different seed bank composition, again, some self-seeding going on there, very patchy, and you have a different response to these tidal flooding and salinity gradients, then potentially it's both who's in the seed bank at what time and how well dispersed they are, and their preference for these tidal wetland environmental gradient conditions. And so moving forward, if you think about, I know this is kind of a heady slide, but if you think about the high marsh right now is invaded by non-natives, the low marsh and mid-marsh is dominated by native species. If you assume that that's because they're both really good at maintaining the seed bank in those plant communities, so there's a differential seed bank going on, resilience, and they also just prefer those areas. Maybe the non-natives they really prefer that wetland, those low marsh areas, and the non-natives really prefer the high marsh areas. Then you would anticipate that both initial establishment and tidal flooding and salinity gradients are kind of driving those compositions. And so I realize this was a lot of information, but as we move through the results, I think it's going to make a lot more sense. Okay, so to evaluate these hypotheses, I went into the field last year, and it was literally this week, 2 years ago, that I was in the field, and I went back to two of the sites I had previously surveyed, the 54-year site and the 6-year site. And my mom graciously took an entire week off of work to help me collect seed bank samples. And so here is a map of the site, and this is a LiDAR elevation map. So green and blue are low areas, and then as you move up, you have yellow and orange, and that's higher elevation areas. So I randomly distributed my native and non-native seed bank samples across both of these sites. And it's also important to note that these sites are hydrologically connected under the road right here. There's a culvert. So at high tide, they are connected. And so this means that the seed bank potentially is really well dispersed, very similar across both sites. And so I collected all of these seed bank samples with the help of my mother, and we also collected soil oxygen reduction potential, salinity, temperature, pH, and we monitored water levels on the site. And so here is a figure sort of showing what that elevation looks like in terms of plant community dominance. And so what I'll point out here is you have the elevation gradient, and then you have these plant communities, reed-canary grass and common rush, lingby sedge and bulrush, and they really zone out into what I would say is called the high marsh, which is dominated by the reed-canary grass and common rush, the mid-marsh, which is really co-dominated by both species, and then the low marsh, which is primarily dominated by lingby sedge and bulrush. And also, what you have here is not just an elevation gradient, by a tidal flooding gradient, which results in lower soil oxygen levels down here, so you have very different growing conditions, and higher soil oxygen levels up here because this area is less frequently flooded. And additionally, the soil down in the low marsh is a little bit saltier than the soil in the high marsh. And this is easily explained if you think about an estuary and the salt wedge that's in the estuary. So salt water is much more dense than fresh water, and so as the tide is coming in, that salt water is at the bottom, and so the lower elevations of the wetlands are going to be more exposed to those higher salinity water conditions, and then that fresh water is floating on top, and so you have fresher conditions in the high marsh. And so not only do you have a tidal flooding gradient, you also have a little bit of a salinity gradient. And so using those data loggers that I had out on the site, monitoring flooding, I was able to summarize on average how frequently and for how long these different zones in the wetland were being flooded. And this is important because I use this data to mimic these conditions in the greenhouse. So the high marsh I found was only flooded for about 1 hour a day, and the mid-marsh was flooded for as long as 3 hours twice a day, and then the low marsh was flooded for as much as 6 hours twice a day. So quite a gradient in flooding. So using those 3 different flooding conditions here, and adding 3 different water salinity conditions, fresh, Oligohaline, and brackish, I created a 3x3 factor study design that I then executed in the greenhouse. And so this, I distributed each of those seed bank samples equally across all of these 9 treatment combinations. And I chose to use the fresh, Oligohaline, and brackish because I wanted to include brackish because even though this is an Oligohaline wetland, in those dry summer months, you do see peaks of brackish coming through. And so because this is a condition that is sort of an extreme condition on the site, this is actually anticipated to be the new normal as sea level rise shifts. And this is like a sea level rise scenario that these sites are going to become brackish. So I thought it would be interesting to see how these seed banks respond to that brackish condition, sort of adding that on. And so I was able to execute this in the greenhouse by using a combination of 27 treatment tubs with their own reservoirs, water reservoirs here, with each having their own salinity combination, with a whole bunch of pumps and timers. And I had 360 treatment pots that were all independent. Okay, so I'm going to show you a video. And so these treatment pots remained independent because we wrapped them in a special fabric that did not allow seed dispersal to occur across. And then I also had drains installed in all of these tubs that did not allow the water level to surpass the pot. So at no point did the water transfer from pot to pot. It was only through that special fabric. And so, give me a second to turn this on. Is my mouse on the screen? Oh, sorry, technical difficulties. I promise it's worth it. Okay. So all of the treatment groups were connected through timers, and so that synchronized the high and low tide conditions across all of them. And this was under 24-hour surveillance. I also was personally, and my interns were in there checking them every day to make sure that the pumps and timers were all working. And then we also monitored salinity of the water and corrected it twice a week, and we monitored the germination weekly. And so weekly monitoring of germination was really important while it was really frequently germinating, because we did not want to have this be a competition study. We wanted to just see what was germinating at what time, what was germinating out of what treatment group, essentially. And so once it was germinating in a large enough size, we removed that, transplanted it, and then grew it out to adult so that I could identify it to species. And overall, we counted over 23,000 seedlings of 43 different species. So there's a lot of germination that occurred. Okay, so jumping right into the data, a subset of each sample that was used in the greenhouse was reserved for direct seed identification. And so this was important. And what that means is literally we went through the soil and identified all of the seeds that were in it. And this was really critical because germination in the greenhouse is going to allow us to identify some of the species, but likely not all of the seeds in the seed bank are actually going to germinate. So to get a really true idea of what the composition was, we needed to really just identify it directly. And so I'm going to show you the data that we came up from that. And the 4 most dominant seeds found in the seed bank were Lingby sedge, bulrush, reed canary grass, and common rush. And that makes sense because this is, these are the plant communities that we were sampling in, so they're doing some self-seeding. Now, this graph shows the native seed bank and the non-native seed bank, and then here we have the average number of seeds out of each seed bank sample. And so right away, what you'll notice is that the non-native portion of each of these seed banks is much greater than the native. So overall, the non-natives are really well distributed. And this means that this was actually composed, both of these were composed mostly of reed canary grass and common rush, so 91% of the non-native seeds found in the non-native seed bank were reed canary grass and common rush, while 87% of the non-native seeds in the native seed bank were reed canary grass and common rush. So very similar compositions across both plant communities. Now, the native species told a little bit of a different story, generally much lower seed numbers across both seed banks. In the native seed bank, the native portion was made up of 53% Lingby sedge and bulrush, while the native portion of the non-native seed bank were only 23% Lingby sedge and bulrush. So this is interesting because it shows that the native seeds species are just not as good at dispersing themselves across both seed banks, and so they have a different composition occurring. Okay, now jumping right into the germination data. So this is going to show you relative germination frequency across all of those treatment combinations. So we have fresh, Oligohaline, and brackish. And then within those I have the high marsh, which is flooded for 1 hour once a day, the mid-marsh, 3 hours twice a day, and the low marsh, 6 hours twice a day. And so those are all pocketed within there. And so the four most common species to germinate out of the seed bank were also reed canary grass, common rush, Lingby sedge, and bulrush. And so I'm going to focus on those 4 species. And so what I want you to notice right away in this graph, and I know it's a lot of information, is just focus on the green bars. And the green, what you'll see is that there is some variability across the flooding and salinity treatments. However, overall, I did not find a significant treatment effect. And so that means that I was equally likely to find the native species germinating in the high freshwater marsh as I was to find them in the low brackish marsh areas. So that was really interesting. And this makes sense if you know what the reference sites look like. So the reference sites, you see Lingby sedge and bulrush growing in the high marsh and growing in the low marsh. So you see them distributing themselves across that gradient. And so that makes sense if you think about it that way. Now focusing on the reed canary grass and common rush, that's the orange and the yellow bars, what you'll notice is that there is actually a significant drop in germination right as you reach the mid-marsh Oligohaline treatment. So you go from high marsh Oligohaline to mid-marsh, and then you see a significant drop in germination. Now this is really interesting because it mirrors what we see happening in the field. These species are dominant in the Oligohaline high marsh, but they drop out significantly as you move into mid and low marsh zones, and we see that really reflected in their germination response. So it's pretty interesting. Now back to this slightly complicated hypothesis figure. If we take these results and we interpret them, what we found was that the native species were showing a really different composition within the native and non-native seed banks. And so they're not really as well distributed. And additionally, we didn't find them responding to these changes in tidal and flooding conditions. And so at the time of restoration, I think initial establishment, who is in the seed bank, if these natives are really abundant in that high marsh zone, they might be able to have a leg up and have a better chance of competing with these non-native species. We kind of infer that from these results. So I think that for the native species, initial establishment and plant competition are going to be really important in determining sort of how they fall out along the zonation in the wetland. For the non-native species, we found that they were really similarly distributed across all of the seed banks, which means that in the low marsh, in the native low marsh, there's a ton of non-native seeds. They're not expressing themselves. And this is because, as we saw in the greenhouse study, they're really showing a differential response to this tidal flooding and salinity gradients. And so this gradient that we create at the time of restoration is really critical to determining kind of where they fall out along that elevation gradient in the wetland, where they develop. And so summarizing these results, reed canary grass and common rush, these common non-natives, are really ubiquitous throughout the seed bank. However, they are suppressed by small increases in salinity and flooding conditions, while the native species, overall very low numbers in the seed bank compared to the non-natives. However, they're not showing any kind of suppression from increases in salinity and flooding. And so this is really interesting, and again, it points to the fact that that initial establishment and competition are probably really critical in determining how these high marshes develop over time. And so maybe whoever gets to the high marsh is winning. Because in these reference wetlands, we do not see dominance of reed canary grass and common rush. We see dominance of bulrush and Lingby sedge. Okay, so overall conclusions. First of all, thank everyone for sitting here with me today. Going through this, I'll just wrap this up in case you fell asleep for a few minutes. For the first study, using an observational study over a chronosequence of sites, I did see that recovery towards reference conditions could be detected. However, especially for plant community composition, this was really tied to the nuances in terms of the changes in tidal flooding and salinity conditions that were developed within these sites. And so we might want to really focus on these nuances as we move forward in these restoration efforts. And then additionally, through an experimental study, I was able to identify some of the mechanisms that are driving these plant community zonation patterns, specifically looking at how seed bank composition and viability under these tidal flooding and salinity gradients is really critical to determining how these plant communities are going to develop on these sites over time. And so I would just like to, again, thank you all for coming. And I really could not have done this with almost every single one of you. And I can't be more, I couldn't be more grateful for your time and your effort. And, you know, Luke, my first wetland intern, showed up today. I haven't seen him in years. And, you know, I just love all of you. Thank you so much. And now I'm ready for really easy questions. Oh, Brian.
Okay. Can you go back just a couple of slides because it'll help me to the conclusions for the second study? Yeah. So I was just curious because you saw that the non-native species germination was suppressed by small increases in salinity and flooding. I was curious if you've had a chance to look at conditions in their native ranges and how that kind of compares to the conditions they're experiencing here, because it could be that like what they're experiencing here is kind of on the cusp of their ability to tolerate, and so those small increases are pushing it over the edge. Again, not needed for this study, but I'm just curious if you've looked into it or not.
That's a good question, Brian. So both of these species are actually hybrids. So they're Eurasian hybrids, three canary grass, and it's a Juncus effusus, subspecies effusus, and so they're pasture grasses. Typically, they're found in these pastures before restoration occurs, and so these kind of pasture holdouts. And biologically, they're both known to sort of tolerate really low salinities, but very similar issues are found sort of even in their home range. They can become invasive, so it's something I definitely could look into more. But
yeah, so they're more essentially terrestrial grasses that can handle, or plants that can handle a little bit of the salt and
the flooding.
But
facultative wet.
They're taking advantage, not really evolved for it. Okay,
yeah.
Cool. Thanks.
Kristen, did you have a really easy question you wanted to ask?
I was curious if you also saw any of the native Juncus effusus mixed in, or if it was all the species effusus.
No, I did not. And I had a botanist with the National Park Service help me try to identify if there was any native out there, and we did not find any. And it's actually it's really, really common because these were pastures. This Juncus effusus, subspecies effusus, is just really abundant in pasture seed banks and in pastures already, and so kind of transitioning to a native, it just can't compete.
Sarah, what's the mechanism that allows the soil to become uncompacted? Seems like a couple of years. I would have thought that those cattle would have really.
Let's see if I can find. I have a slide. Is that it? Yeah. So this is a really good question. So this is a picture of, a graph of the soil bulk density versus the organic matter. And what I want you to notice is that there was actually a strong correlation between organic matter content and bulk density. And so what I think is happening on these sites, and I'm focusing on the surface soil. You have to remember this is the surface soil. And so over time, first you have the water come in, kind of fluffing the soil, if you will. That's probably not a technical term. The water's coming in and flooding it, and then you have sort of organic matter accumulating right away, because as you reduce the oxygen levels in the soil, the carbon is going to accumulate, or the organic matter is going to accumulate. And so I think that we see a really strong relationship between soil bulk density and organic matter in these sites, because that's probably one of the major mechanisms. That's my guess.
And is that sort of opposite of what you see in dry land range?
You don't see that recovery as quickly, and I think it is because you just don't have the productivity that you have in these, wetlands. And then they're Oligohaline, and so they're even more productive because they're not super salty. They're like not a salt marsh, you know. Did you have a question? No?
Me?
Yeah.
Yeah, I have a quick one. I know you said you saw no trends in the nutrients. Do you have any thoughts or ideas about what's going on with, or, just some of your, because you know that area so well, what you think is going on with nutrients, nitrogen and phosphorus there?
Yeah, I found the lack of trend really interesting, and I found it really interesting because when I did sort of a pilot study, I went out and measured water quality on the pasture site, and I found that the nitrogen and phosphorus levels in like little ponds on the pasture site are really high. And so I'm wondering if they're just I'm wondering if they are, I don't know. So I know that at some point there was some elevated nutrients in there, and my data is not necessarily showing that trend, and so I'm wondering, maybe there's just really a lot of natural variability in nutrients among these sites and within this area. As any good scientist, I would say, I think that if I was going to do it again, I would go and collect more samples just to confirm that there is natural variability and I didn't just miss the trend. You know what I mean? But hopefully I don't have to do that to finish my
Quick question that might be of interest to people. To point out, outstanding work, I've seen some field work. So one thing that you found was that the high marsh sites aren't coming back to reference conditions as quickly as the low marsh sites. And so I wonder if you could just, if we're going to keep doing these kind of restoration attempts, and hopefully we are, what could we do to try to accelerate the high marsh restoration?
It's a really good question. I've thought a lot about that because
Yeah, me too, because my site's
it's really critical to think about. I think that the easy answer would be to lower the high marsh to make it, but the problem with, I'm not advocating for removing the high marsh because, if we think about sea level rise and climate change, and of course, sediment distribution within the site, how long would it take for those sites to gain that elevation again? And we don't want to lose those critical high marsh habitats. And so, that's the easy answer. I wouldn't go down that road. So, I think that we might need to consider how water is getting to these high marsh areas and maybe doing some, groundwater monitoring to make sure that they are getting actually as much water as reference marshes, and maybe if they're not, we need to sort of try to think of how we might engineer more water to be getting into the high marsh zone if it is lower, like a lower amount of water infiltrating into the soil than the reference sites. Additionally, I think we might need to remove at least some of the top layer of soil just to remove that non-native seed bank, particularly in sites that are already heavily invaded with common rush and canary grass, because that seed bank will stay for like 70 years, and maybe do some seeding in those areas with native or planting. So more active approach in that high marsh zone.
Do you have a sense of what is driving the variability among your reference wetlands? Because you have a really wide range of values in your reference wetlands, which drives your early recovery window for the restored sites, right? So if all the reference wetlands were sort of closer together, that would make the bar higher to achieve restored conditions, right? So can you talk about, I don't know, are there differences in the amount of area between high and low marsh in these different sites, or just sort of speculate about what's going on?
Yeah, I'm gonna just move back to, help me, just really quickly. Oops, I keep wanting to use that mouse. Sorry, just 1 second. Let me get a map so I can look at this more clearly. Okay. So my reference sites were ranging from Daggett Point here, sort of in Youngs Bay proper, here near the Cowlitz pasture. And this site, which is CT in all of my graphs, is adjacent to the pasture site. So this site in particular, it is not diked and drained. However, I do think that once in a while, probably a little bit more frequently than the other sites, it's going to have cattle wandering onto it. And so we do have sort of that potential cattle influence on this reference site. And then this site is an island, which in itself, the plant community was really similar. You know, all of these sites were actually fairly similar. So if you looked, when I was looking at comparing them and whether they were significantly different from one another, they were sometimes significantly different, but for different variables. So there wasn't necessarily a trend. You know what I mean? So I was trying to piece that out. And then we have up here, really deep into the watershed, Cooperage Slough. And so this, and this site is, you know, I was like, well, maybe it's got a lot more freshwater conditions because it's so deep into the site. But, you know, honestly, I could not find a good reason for the differences, you know, sort of using the data that I had at hand. And I did feel like it was a good range of sites to compare to because clearly even reference conditions are going to vary. I think that in particular, though, this site did have more non-native species, and I do think it was because it was so adjacent to the pasture site. Yeah.
You said cattle maybe wander onto your reference site occasionally. Aren't there a lot of elk there that move around?
So that's very true. So there are elk herds that move through the area. However, they're really restricted because of roads and like dikes and levees. So there's actually, I don't think that they get into this site because there's actually a channel that sort of separates this site from the mainland here, and then this is all actively cattle grazing, and so they sort of restrict that. And then this site, I don't know, Rudy, do you think that there's a lot of elk on your site?
Oh yes, there are a lot.
Okay, so there's potentially some elk impacts there. Reference site, unlikely, the island, they're probably not going to get a lot of elk grazing here. And then this site, it's possible that there were elk there. I didn't see any, and it is surrounded by development, so it's hard to say. I didn't see a lot of evidence of grazing on any of the reference sites, so I should make that really clear. Even the one that was next to the pasture.
Just a quick note, so elk do manage to find their way all over the landscape down there. Fences and dikes
And they swim,
waterways,
so.
no barrier to outroads about elk.
Thank you. Oh.
So, Burgess has some work showing that the type of competition that drives, like, which plants are successful depends on the nutrient levels. So it can be competition for nutrients or competition for light, depending on terrestrial inputs, usually of nutrients. And that therefore there can be a high variability in which plants are there and successful in perpetuating seeding. So do you think if you looked at the variation in nutrients, that I know you didn't see significant differences, but that doesn't mean they weren't variable.
Right.
And that could help explain some of your variation in how plants are doing, which plants are doing well.
I did look at that. So I did look at that, and I found that there was a slight positive correlation with the amount of nitrogen in the soil and the amount of native species richness. However, it was a really slight correlation, and you know there were a lot of other things that were correlated as well. Nitrogen was also really, really strongly correlated to organic matter content, which makes sense. Yeah, so it's definitely something that makes sense. Oh, Brian.
I've got the best question. What's your favorite wetland plant?
Oh, that is a really hard question. I love them all. Katrina, what do you think my favorite wetland plant is? Do you remember any of them?
Yes.
I know Katrina's favorite wetland plant was bulrush because she would like to make crafts out of it. I really can't call favorites.
I'll take the
And then if I say if it's an invasive species, then I'm probably a bad person. So.
That's like asking who's your favorite child. Come on.
Yes.
Yes. Yes, husband.
Who collected the LiDAR data you used?
Your company collected the LiDAR data.
I think that was me personally.
Oh, you personally collected. Well, I clearly didn't pay you to do it, so that was a coincidence. Well, if we have come to a close of questions, I just want to say that, again, I am really, really grateful to everyone for coming today and for all your help through the years, and think really positive thoughts as I'm in here with the committee by myself. And please help yourself yourself to some treats before you leave. Brian Turner's wife, Crystal, made carrot cake, wetland-themed cupcakes.